Hello, today I'm going to be talking to you about how the brain responds to delivering drugs across the blood-brain barrier using a rapid short pulse sequence of focused ultrasound. Now, traditionally, people use long pulses of ultrasound to deliver drugs across the blood-brain barrier. What we're doing is emitting a sequence of rapid short pulses that we call RaSP. And the idea here is that we're trying to distribute the microbubble activity within the vasculature so that we get a more uniform drug delivery inside of the brain. Now, when we tested this in vivo, this is exactly what we saw. A more uniform delivery inside of our brain, so the drug is reaching more of the cells, but also the blood-brain barrier is closing within just 10 minutes rather than being open for hours and hours. And if it's open for hours and hours, this also means that not only your drug is going to go across, but also other toxic proteins are going to be able to enter the brain as well. So, do neurotoxic proteins enter the brain? And I'm specifically looking here at albumin and immunoglobulin. These are the most common proteins that are normally in the blood and not in the brain. If they enter the brain, they cause neurotoxic effects, the DNA fragmentation inside neurons, and this causes neuronal death and also the activation of immune cells as well. So I'm looking at albumin and immunoglobulin. Immunoglobulin is a slightly larger protein, almost double the size of albumin. Now, in terms of albumin, what we see at an initial time point, 0 minutes, is that with the RaSP sequence, we get 4.7-fold less extravasation into the brain compared to the long pulses. And at 10 and 20 minutes, we get none at all. So whatever is getting into the brain at an initial time point is being excreted within a very short amount of time. With the immunoglobulin, the much larger protein, we see none at all across all time points with the RaSP sequence. So our size threshold of compounds that enter the brain with this RaSP sequence at this pressure is somewhere between albumin and immunoglobulin. And overall, we see less of these neurotoxic proteins inside of the brain with the RaSP sequence. Now, in terms of the glial cells, the immune cells of the brain, are they responding or not to what we're doing? I'm looking here at microglia and astrocytes. These immune cells of the brain, they are both involved in removing any unwanted compounds from the brain, such as our model drug. But what they also do is, if there's inflammation in the brain, they become activated. And what they do is they change their morphology. So in terms of the microglia, what I see is in blue with the RaSP sequence, you'll be able to see that across 48 hours, we get a lot less of the microglia uptake in our model drug in these brains. In terms of the morphology, when microglia are resting, they have a branched state, branched morphology, ramified. When they're activated, they retract these branches and become a lot more rounded so that they can be recruited to a site of damage in the brain very quickly and they're very mobile. Now, from my staining, what I see here, you're looking in green at the microglia stain and in red is the model drug. Now, with the rapid short pulses, the RaSP, you'll see that these microglia are ramified and branched. Whilst with the long pulses, all of the microglia that are uptaking our model drug are more rounded. So their form, their morphology is an activated state. With the astrocytes, in a similar way, we see a lot less uptake with the RaSP sequence. You'll see this in blue within astrocyte cells of our model drug. In terms of their morphology, they're different from the microglia. When they become reactive, the astrocytes become bigger. Both the cell body and also the branches become thicker, and that's a way of recognizing the reactive state of the astrocytes. They occupy a larger area inside of the brain. So if we quantify the area covered by these cells, we actually see that with the RaSP sequence, this is lower. So it indicates that there's less of these reactive astrocytes inside of the brain compared to the long pulses. So overall, we're getting fewer neurotoxic proteins inside of the brain with RaSP. We're getting less of this removal from the immune cells and less of an immune response in general, it seems, from the morphology of the cells. So what does this mean? Does it mean that with the RaSP sequence, we're causing less disruption to the brain, which could give us more confidence in being able to give patients multiple treatments with this technology, possibly? So our next steps are to look deeper into the finer details of what's happening inside of the brain, brain cells, but also at the level of the barrier, so that we can understand better what type of disruption we're causing with the RaSP sequence and how this is giving us less safety concerns. So with that, I'd like to acknowledge everyone who's helped with this work and funded it. And I'd also thank you very much for listening. I'll take any questions. Thank you.